So in this lecture, we're going to explore the idea of nature versus nurture and look at how changes in how we do meiosis from the normal leads to more genetic variation or potentially genetic diseases. So with heredity, we have this idea of a chromosomal theory of inheritance. All of our alleles are found on chromosomes, and chromosomes, if you remember from our previous unit, are just packages of DNA that are wound around proteins called histones, and they coil very tightly around each other to make these discrete units. And every single gene is found at its own specific loci or position within the chromosome itself. So this particular example that I just squared on the right, that is the locus or the location for the flower color gene on this particular chromosome. And you can have chromosome 1 and chromosome 2 as the homologous pair. And it's these chromosomes that are the basis for Mendel's laws of inheritance. So his law of segregation, remember it said that 2 alleles for each gene separate when the gametes are formed. So I have my 2 homologous pairs here, but they're going to be separated into their own cells through meiosis. The cells will go from diploid to haploid. Now, there are some circumstances where that doesn't occur properly, and you may end up with both of these moving into one cell and none moving into the other, and we'll talk about that too. You also have the law of independent assortment, where the alleles of genes on non-homologous chromosomes mix independently of other chromosomes. Basically, you don't have all of the blue ones lining up on one side and all of the red ones lining up on the other side during metaphase. They will randomly assort themselves into any particular order. All of these things just increase the possibility of genetic diversity. We don't necessarily want to have every single time we do meiosis to have the same mix of all of our DNA. We would prefer this more random mixing of our chromosomes. So when our chromosomes do not follow this law of segregation perfectly, we end up with something called non-disjunction. This is a failure of our homologous chromosomes to separate during anaphase. Instead of them pulling apart like this top picture here, they end up sticking together, and you end up with too many chromosomes in one cell and too few in the other cell, or potentially none at all. So plants don't seem to have a problem with this. They are often what we call polyploid, where they'll have more than 2 copies of every single chromosome in their cells. So some are triploid, like bananas and watermelon. Some are tetraploid, like blackberries and peanuts. And then you even have circumstances where you have wheat, which is hexaploid. They have 6 copies of every single one of their chromosomes, or strawberries that are octoploid. We found that a lot of times this polyploidy was a result of human intervention. As we were selectively altering and breeding these plants, they ended up getting more of these chromosomes through our breeding. Now with humans, we don't tend to like this non-disjunction. It tends to have more severe consequences for animals when this happens during meiosis. You'll either have a developmental disorder if you have too many chromosomes in the cell that was chosen to be you, or you're going to see miscarriage occurring. So if a cell, if the gamete that gets fertilized is one that didn't have enough chromosomes, you're going to end up with a organism that can't even make it through gestation. They'll die while they're in the uterus still. So there's 2 opposites here. You can have aneuploidy for animals or humans. And this is where you have 1 too few copies of your chromosomes. You're 1N. You only have 1 copy of the chromosome in that particular gamete. And there are some disorders that are linked with this. If it's an autosomal chromosome, chromosomes number 1 through 22, often the organism won't even be born. There'll be a miscarriage. However, when we're dealing with our sex chromosomes X and Y, there's not too many chromosomes on there that we can't live without if we only have one copy, because remember, we selectively inactivate our X's so that essentially all females only live off of one X and all males only have one X. So only getting one copy of these chromosomes is not necessarily a big deal. The example is monosomy X, or Turner syndrome. This is a condition where you will have only one X chromosome, no Y, no second X. And these people will develop as phenotypically female. However, they'll be sterile. Sometimes they can have intellectual disabilities, but most often they do not. They tend to have this very distinctive neck shape that you can see in the woman there. You don't often see monosomy Y because there are genes on the X chromosome that are required for life. You cannot live without at least one X chromosome, but you can live without Y chromosomes. The other side of this is when you get too many copies. The most common example in humans is trisomy, where we have one too many copies, so we're 3N for that particular chromosome, 3 copies of that chromosome. And the most common one, trisomy in the human population, is trisomy 21, or Down syndrome, where we have 3 copies of the 21st chromosome. And these individuals are born with a very distinctive facial features. They're often a little bit shorter than normal. Their intellectual capacities can range from needing constant care and support to being able to function on their own and have their own families and careers. So with trisomy 21, it's a broad spectrum of symptoms. This seems to happen more frequently when the male and female that are reproducing are over the age of 40, and I believe for males it's over the age of 50. As you get older, your ability to do meiosis faithfully will start to degrade. So you'll experience more instances of non-disjunction, more chances of having an offspring with something like trisomy 21. Now, we can also have structural changes to our chromosomes themselves. We learned about these a little bit when we talked about mutations. We can delete a portion of our genes, or of our chromosome. Like this first example here, we had a D portion and now it's gone. We can duplicate where we had a B and C and now we have B and C copied twice. We can invert where the order of particular sections of our chromosomes can switch and invert on itself, or we can have translocations. Now, this looks a lot like crossing over, but these are on non-homologous chromosomes. It's 2 chromosomes that have completely different genes on them, switching information as if they were trying to perform crossing over. Now, these can sometimes have no consequences at all, especially if we're talking about like plants and they have a duplication. Once again, plants can live with, a polyploidy state of sometimes having 8 copies of their chromosomes. So having an extra copy of a couple genes isn't necessarily going to affect them negatively. However, you have a problem with maintaining faithful sequences of DNA when we're moving things around on our chromosomes. We can end up cutting a gene halfway through, and now the gene has part of its sequence on one portion of the chromosome and the other sequence at a different loci. So there's no way for transcription and translation to occur because the gene is separated from its other half. You can also have a gene that gets moved properly, but all of its upstream regulatory sequences are gone. So when we talked about things like the lac operon, you're going to have all of those operators and the regulatory sequences maybe are separated or gone. So now we don't know when to copy our lac operon. We don't know when to express it. We don't know when to not express it. So often, these structural mutations in animal cells cause significant problems because we're not cutting the chromosomes in a good position. An example of this is Cri du Chat. It's a deletion in chromosome 5. So they have the situation seen in A here in chromosome 5, where you end up missing some of your genes or a portion of your chromosome. And these individuals are born with severe intellectual abilities. They often have a very small head, and they have the disease has the name that it does because the sounds or the cries that these babies will make often sounds like a kitten that's trying to meow. Now, this particular disorder is pretty much fatal in infancy. Very few even survive into early childhood. We talked a little bit about our dihybrid crosses, where we were looking at two genes at the same time, like the round and wrinkled genes, or the green and smooth genes, things like that. So with those dihybrid crosses, they're due to unlinked genes, genes that are not always seemingly stuck together. And they result in something called recombinant types. These are phenotypes that are expressed in offspring that you can't see in the parents. So if you take a round and green pea and you mix it with a wrinkled yellow pea, you can end up with a wrinkled green pea. Neither parent had that phenotype, but it is expressed as a recombinant type in the offspring. Now, this is not the same thing as a linked gene. Linked genes are those that are really close together on a chromosome, so they are essentially always inherited together. In the example here on the right, these particular chromosomes, the G gene and the A gene, are far apart from each other on the chromosome. So most likely these genes are not going to be linked. Crossing over may occur and actually separate them. However, in this example on the right-hand side of that image, they're very close together. The likelihood of crossing over, separating those particular genes is very, very low, because as the genes get closer and closer together, it's less likely that crossing over is going to occur between them. And the only way that these types of closely associated genes can become unlinked is through crossing over. Now, Alfred H. Sturtevant, he was a student of T.H. Morgan who worked with those fruit flies and discovered chromosomes. His student, Alfred, decided he was going to make a genetic map. Once they kind of discovered that some genes were linked and some weren't, he wanted to do more research into that. So he created a list of all the genetic loci of a particular chromosome, and he called it a genetic map. He found that the farther apart that two particular genes were on a chromosome, the more likely they were to be separated from each other. The closer they were to each other, the less likely they were to be separated. And through that, they discovered crossing over. So if you have a higher chance of crossing over between two genes, we call that a higher recombination frequency. We're increasing diversity, creating a recombination because we're separating genes from each other. And he said that the distance between different loci on his maps were called map units. And one map unit, it corresponds to 1% of a recombination frequency. So you can actually then calculate the probability or percentage of two genes being separated by crossing over. The last thing we're going to talk about is environmental effects on heredity. We talked a little bit about epigenetics when we were discussing DNA and how sometimes we can coil more tightly around our histones or less tightly. We can have methyl groups added that makes this coiling or uncoiling happen, and that affects our ability to copy and translate certain genes within our chromosomes. Well, not only does that happen, but we have the environment affecting whether or not particular genes are transcribed or expressed. It's this whole idea of nature versus nurture. If we were only determined by our nature, then we would only see our phenotypes because of our genes. If we all had a specific type of gene, we would all show that phenotype. If we are determined by our nurture, then our genes have nothing to do with who we are. It's all about our environment and how we were raised. And we're finding more and more that reality is a mix of the two. When we first really discovered genes and genetics and DNA, a lot of the idea of nurture went out the window. And there was a lot of studies trying to determine if everything about us was linked to our genes. There must be a gene for everything. But as our tests have become more and more specific and our understanding of genetics is becoming deeper, we're finding that it really is a mix of nature and nurture. Our genes really do affect who we are, but then who we are is also created by our environment because our environment is changing how we express those genes. That's called phenotypic plasticity. You can have one genotype that can produce more than one phenotype depending on the environmental conditions. Now, this kind of makes sense, and we've already talked about it when you consider the lac operon. The lac operon has a change in how it's expressed depending on what its environment is. If it has a lot of lactose, it's going to be expressed very differently than when there's no lactose. It's going to be expressed less strongly if there's no camp and more strongly when there is camp. So that is already an instance of environmental effects on that particular gene expression. That's an example of phenotypic plasticity. So here's some examples. And we're going to start with one that's really common. Let's say it's spring and it's finally starting to get warmer. You're going to go out to the beach all the time and you notice you get a really nice tan. You're no longer as pale as you used to be. Well, that's because the amount of UV light that you are exposed to affects how your skin produces melanin. The melanocytes, or the specific cells that produce melanin within your skin, are going to have to make more melanin to protect you from the increase in UV rays. So as a result, if you have more melanin, you're going to have a darker skin tone, and we call that like tanning. Another example is hydrangeas. You can plant a hydrangea in a soil with a different pH or a different acidity level and get a different color flower. If you plant the plant in an acidic soil, you're going to see blue flowers. If you plant it in a soil with a pH of 7 or higher, they're going to be pink. And you even see a gradation between those different acidities. You can get a deeper color or a lighter color depending on that pH. Another example that you're probably really familiar with is the changing of the fur color of Arctic animals and birds. In the summer, they will all look darker or brown or have a mottled fur color, and in the winter they all turn white. Well, this is because the genes that they're expressing change depending on how much light is available. We call that the photoperiod. As the days get significantly shorter, that is a signal that winter is coming. And in these areas, they tend to have more snow in the winter. So the animals will molt, they will lose their particular fur or feathers and regrow ones that have a different coloration pattern because we're not expressing the same genes. Another example that's a really cute one is Siamese cats. There's a gene that creates a temperature-sensitive enzyme called tyrosinase. And this gene is deactivated at high temperatures. When that happens, they cannot make melanin anymore. And melanin is the pigment that makes skin or hair or fur darker. So when you look at a body in general, it tends to be the warmest in its center of mass. In its chest, in its abdomen, you have more mass there, it's going to be a lot warmer, and you get colder and colder as you move towards your extremities. Your fingers, your nose, your ears, those are going to be so much colder than your armpits or your stomach. So with these cats, when they're born, they all look the same. They're all one color. But within a few hours or days after birth, the temperature change between their extremities, their paws, their ears, their face, their tail, is going to be so different from that of their core body that it turns a different color. The core body can't make melanin, so it's a lighter color. The colder temperatures of the ears and the paws can make melanin, so they become darker. And the last one that we're going to talk about, I believe I've mentioned before, is determining the sex of reptiles based on the temperature of incubation. This happens a lot with alligators, with lizards, with turtles. In turtles in particular, they have a really fascinating pattern. You can see turtles where the extremes of temperatures produce one gender and moderate temperatures produce the other. So you'll have the really hot and really cold producing males and the intermediate temperatures producing females, or vice versa. You can also see one where it's just one particular separation between sexes. So hot temperatures produce females, cold temperatures produce males, or the opposite. And this is because the genes are going to be expressed differently at different temperatures. The genes that produce male hormones will be expressed at certain temperatures, and the genes that express female hormones will be expressed at the other temperatures. And that's all that I have for you guys today. If you have any questions, let me know.